I think we have a lesser crowd today. One thing is quite a few people from my lab has, I mean, some of them have gone to South Korea for some training, and it's pretty late for them, and so they won't be joining in. There's one gone to Haeshan who was also coordinating this. He's in Hong Kong right now, so I think because some people have, gone for training purposes and, research training, some from our lab have not joined today. And of course, the other reason is the YouTube channel we are having. A lot of people are viewing all the talks, not just once, but several times. So when you have the option of, watching the talk at your own pace, I think people tend to prefer that better possibly. So Diniti, do you want me to give it a few more minutes, or?
Maybe I can.
Yeah, you're muted, Diniti. Yes, somebody else is.
Yeah, me and Lisa are both in the same room, that's why.
Okay, but I can hear you, yes, through Lisa's mic, yes.
I was thinking maybe we can give another couple of minutes.
Yes, why not? Yeah, why not do that? So thank you, Diniti, and everybody else for, really doing this. It means a lot to us, and I think everybody here in Sri Lanka are appreciating it. So thanks a lot.
It's our pleasure. Yes. And thank you so much for the support all throughout from your team as well.
Yes, as I said, quite a few of them are not in Sri Lanka, right? I mean, they have to go here and there for training purposes. So we'll give 1 more minute and then we can start. But in the meantime, I'll just share my screen to tell you all about this meeting that is happening in Kolkata, India. So Sri Lankans, I mean, they are offering some travel grants as well to support travel expenses. So I think it's a good idea for anybody who is interested. I mean, I think we showed this at the beginning of the seminar series, but since this is the last one, and you have a much better idea of the human cell atlas, and also how single cell sequencing can be used to define how all these things work and the power of it. And I think you'll be more interested in attending this, and Kolkata is not too far away, and I think many can sort of consider going here, especially when they are offering certain travel grants. So we do have a few more people joining in, Diniti, and as I said, quite a few members of our team have gone away. So we will stop sharing and introduce today's speakers. So the first speaker is Lisa Dratva. Lisa, I don't know if I Dratva. I hope I pronounced your name properly.
Dratva is fine. Yes, yeah, exactly.
Yes, thank you. So she's been trained as an engineer at EPFL Switzerland, and her background lies in analysis of methods of development of single-cell data and cellular atlases. And for her master's thesis, she built deep learning model to predict gene expression at the Church Lab in Harvard Medical School, and has multiple experiences in industry with big pharma and with small startups. So quite a wide range of experience here. And since joining Sarah Takeman's lab at the Wellcome Sanger Institute, her research has been focused on understanding human immune responses to COVID-19. So, and especially T cells. So that's a really exciting talk that I'm also quite interested in. Not that I'm not interested in others, but you know, specifically interested in this. And and she's applying statistical and machine learning to a single cell gene expression in human repertoire data. Our next speaker is Kazumasa Kanemaru. I don't know if I pronounced that right. And Kazumasa completed his medical degree in 2012 and PhD in 2016 at the University of Tsukuba, Japan. During his PhD, he focused on the functional analysis of immunoreceptors using mouse models and cellular assays. And Kazumasa joined the Takeman lab in October 2020 and has been focusing on data generation analysis of the human heart using single cell and spatial multi-omics technologies. So thank you so much, both of you, for being there today. And over to you, Lisa, to start off.
Great, thank you so much.
I hope you have got the host.
Not yet, I think you have to enable it.
Okay, let me see. Okay, how do we make you the host? Okay, make host. Okay. Okay, I think I made you the host.
Yeah, it works. Thank you.
Okay, great. Thank you.
Okay, can you everyone see this and hear me all right?
Yes, yes.
Okay, I'm just going to turn off meeting controls. Great. Thank you so much for the nice introduction. I'll tell you today something a bit about understanding COVID-19 using single-cell genomics. And as was said, I am Lisa Dratva. I'm currently a PhD student in Sarah Teichmann's group here at the Wellcome Sanger Institute in the UK. And yeah, thanks so much for joining today. So the aim of this talk will be to present you with the results on our analysis in the COVID-19 challenge study. So this manuscript we have recently uploaded on medRxiv. That was in April, and it is called Human SARS-CoV-2 Challenge Resolves Local and Systemic Response Dynamics. So feel free to also look that one up if you want to go more in depth on the results that I will talk about today. Now, just as a little introduction, single-cell biology is, to me, a new frontier in biology. It helps us to ask questions such as, what determines cell identity? How do cells assemble to form tissues and organs? And what is the cellular basis of disease? Now, once we have a better understanding of how different cell types come together to form such tissues and organs, we can also investigate, for instance, which cell types are responsible in the cases of disease and so on. And my PI, Sarah Teichmann, she has been involved in founding the Human Cell Atlas a few years ago, whose mission it is to map every cell type in the human body. And today's work will also integrate into this bigger project. Now, how do we go about understanding COVID-19? What was done basically since the onset of the pandemic is that usually you would be able to take samples from COVID patients that were, for instance, admitted to the hospital or that at some time had a positive PCR test. What you would then do is you would, for instance, take a blood draw from them, and you would then analyze the different genes expressed in the different cells that were present in these samples of COVID-19 patients. And this was done ever since the pandemic started, for instance, by our lab in collaboration with labs in Newcastle and UCL London. However, this kind of approach, it suffers from a big limitation. And this is that every time we just take a sample of a COVID patient that just happened to be in the hospital with COVID, all we get is one static snapshot of the disease. We don't really have a baseline sample of that same person that would allow us to determine what are the changes that are now potentially due to the disease. This is where human challenge studies come in. Because what you can do with a human challenge study is you can give a controlled set of individuals an exact dose of, for instance, SARS-CoV-2. And then you can go on to explore things such as protection and variability that differs between individuals. You can also investigate things such as the cellular events that come before symptoms or before a positive PCR test. And importantly, you can create a very detailed timeline of this infection in humans. So this whole body of work, we hope, will serve as a Rosetta Stone of SARS-CoV-2 infection in humans. And just to say, basically, to be able to participate in this study, only people between the ages of 20 and 30 years were allowed, and all of these people had to have never had COVID before, and they were also not vaccinated. So by doing this, we hoped to have a sample of individuals that are relatively similar, and so that we would be able to really explore these differences between them. The main questions that I'm going to talk about today are: what happens to the epithelium at the site of infection? And how does the myeloid compartment change over time? And then finally, can we characterize the adaptive immune response to a viral infection such as SARS-CoV-2? And just to start off, here's to acknowledge the two first authors of this manuscript, which are Kaylee Warlock, currently PhD student that actually just defended at the Nikolic group in UCL. She performed all the wet lab handling of samples and so on for the study. And Rick Lindeboom, who headed the data analysis for this study and was until last year a postdoc in the Teichmann group. Now, the model for our human challenge experiment looked like this. Every volunteer was sampled at regular intervals, and importantly was sampled once before being given the virus, so that for every individual we have a very clean controlled data that we can compare all the future samples to. And we took from the individuals at the different time points blood samples as well as nasal swabs. And then all of these were frozen because there were so many samples, and then thawed when it was time to process them. And they were processed on the 10x machine to yield single cells, and we also sequenced their gene expression, their T cell receptors and B cell receptors. Interestingly, even though we had 16 individuals from common backgrounds, only 6 of them went on to develop COVID-19 with symptoms and positive PCR tests. Actually, 7 of them never got COVID and also never had a positive PCR test, and 3 of them were somewhere in the middle, so some in-between phenotype. To come to the main question, what actually happens inside the nose, so at the epithelium at the site of infection? To check this, we aggregated all the single cells that we had from all the individuals, so that totaled in 234,000 cells, and we annotated them with very fine-grained cell states, in total 73 cell states. And you can see we have a really big compartment of ciliated cells, we have several immune cell niches as well, and then we have some other cells such as goblet cells, secretory cells, and so on that you would find in the nose. And as you can also see on the right, certain regions of this UMAP are kind of exclusive to sustained infected people. And you can also see that there is a kind of time progression across this, this representation, which I will talk more about now. When we look at the immune infiltration in the nose, this graph gives us quite some information on this. So here a red dot means that the given cell type increased with time, and a blue dot means that it decreased with time in abundance. And as you can see in the sustained infected people, so the ones with COVID, around day 5, we have quite a clear increase of all these immune cells in the nose. Interestingly, in the transiently infected people, this already happens at day 1, so they are clearly different. And you can also see that the abortive infection group never actually had an immune infiltration in the nose. So they never mounted a proper response over several days. And we also looked whether we could quantify the number of SARS-CoV-2 reads in the cells of the nose. And what you can see here nicely on the left is that the peak in SARS-CoV-2 reads in the nose was detected around 1 week after inoculation. And we were able to break this down by infected cell type, or cell type with viral reads. And you can see that especially the green one is very pronounced. So these are the ciliated cells. They clearly show up as the main producers of SARS-CoV-2 virions in the nose. Interestingly, we also have CD8 T cells that had a lot of viral reads detected in them. What I'm not showing here is that we were able to quantify whether these were productive or not productive infections of cells. And we could see that only the ciliated cells were actually the ones producing virions. The CD8 T cells wouldn't really produce virions in the same way. On the right-hand side, you can see that the ciliated cells we could further distinguish into kind of normal ciliated cells with potentially slight infection or low viral reads, and a hyperinfected ciliated subtype that really had enormous amounts of virus compared to all other cell types. To take this apart a bit, we looked further and in more detail at the ciliated cell states, and we actually could see that there are 5 distinct states. So on the one hand, we have the normal ciliated cells, then we have ciliated cells which are interferon stimulated. Then we have the ones that are also hyperinfected, followed by the ones that are slightly infected and have MHC class II proteins upregulated. And we have a 5th type, which also has the acute phase response genes upregulated. And just for reference, MHC class II molecules are important they're usually only expressed in professional antigen presenting cells. So we are not quite sure what this means in this context. This is an unexpected finding. And if we look at how these ciliated cell states come about, it's actually, they aren't all present at the same time or throughout. Instead, we have first the acute phase response ciliated cells peaking around day 1 in the nose. Then we have the interferon stimulated population, which grows and peaks around day 5. And then the other 2 populations we only see from like day 5 until day 10. So importantly, we would not have picked up on this had we only looked at a particular sample, and we wouldn't have this longitudinal data. To our next question, what were the changes in the myeloid compartment? For this, we looked to the blood data that we have collected, which accumulated to 371,000 cells. And the big number of 129 cell stage. So we have very detailed and fine-grained cell annotation. Again, as you can see, all the major immune compartments were found across our data as well. And in this plot on the right, you can again appreciate that, interestingly, here we have a few blue dots. And this means again that these cell types decrease in abundance with time. So we have quite early on a decrease in the dendritic cells as well as the inflammatory monocytes. And this basically tells us they left the blood, presumably they go somewhere in the tissue, and we presume that they would so go to the nasal tissue or where the infection is happening to presumably take part in the initial response to this infection. Now, we also found that interferon signaling, which basically tells the body that an inflammation is ongoing or is being mounted, preceded in the systemic, in the system instead of the local response. So what this means is we first detected it in blood around day 3, which is this red arrow on the right, and it was only then being followed by the local response in the nose. This was quite surprising to us because we thought you would first have the signaling in the nose and then in the blood, but actually it turns out to be the inverse. And to come to our last question about the adaptive immune response, we actually found an interesting novel activated T cell state, which was restricted to the sustained infections, so the people with COVID-19 and positive PCR tests. These cells appeared first around 1 week after exposure and stayed for another 1 to 2 weeks and then disappeared again. So they were clonally expanding T cells, and interestingly, we did quite some analyses on their T cell receptors, and we found that there were shared TCR motifs among COVID-19 patients, which were missing from healthy controls. And we also were able to show that the T cells that are these novel activated T cells, they have SARS-CoV-2 specific receptors, as we found by comparing this to a antigen specificity database. And we could see that indeed these activated T cells were significantly enriched for SARS-CoV-2 specific sequences. And in line with this work, we also produced a tool called Cell2TCR. Basically, this is a two-step tool to bring you from a pool of T cells to a group of a smaller subset of T cells that are sharing important T cell receptor sequences. The way it works is that first you would apply a transcriptomic annotation algorithm, such as Cell Typist, to figure out what are the different cell states that you have in your data. Then you can zero in on these activated T cells, which I've just described, and you can see whether among the activated T cells of different patients, of, for instance, a same disease, you find shared motifs in the T cell receptors. Once you have found such groups, which is what we found in COVID, you can assume that these are likely disease-specific, especially if they are missing in a control group. And we applied this in our data, and we found that we have over 200 shared or public TCR motifs among COVID-19 patients. And I've shown here the most common motif on the left, and you can see it has two parts to it, the TCR alpha and the TCR beta chain. And yeah, if you want, you can try out this tool on your own data, on your own T cell data, and we would be happy for any feedback on how this worked out for you. And yeah, here just to summarize, we characterize the highly dynamic cell states by compartment, infection group, and time point in the challenge study. We found that a strong interferon response in the blood preceded the one in the nasal pharynx. And we identified a novel set of activated T cells, which are clonally expanded. They peak 10 days after infection, and they carry public SARS-CoV-2 specific motifs. And yeah, we hope that you may also find this interesting and potentially compare your own samples of infection to this, so that our data may serve as a sort of Rosetta Stone, as I said before. Thanks a lot for your time, and also thank you to all my collaborators on this. And if you have any questions, I'll be happy to take some of them now. Thank you.
Thank you very much, Lisa. That was very exciting results and great presentation. So anyone has any questions from the audience? We have a comment saying very interesting. Thank you, Bayan.
Okay. Thank you. Yeah.
So we will go to the next talk. In the meantime, if you have any questions for Lisa, you can type it in the chat and she will answer for you. Thank you again, Lisa. And now let us hear from Kazumasa on his work. Thank you, Kazumasa, for joining us today.
Yeah, thank you for organizing this seminar series. I can't share my screen, so.
Yeah.
Oh yeah, now, yes, now we can. Yeah, hello. Thank you again, and I'm delighted to be one of the presenters of this series. And I hope you can see the slides. I'm a postdoc at Sarah Tahiman's group, and I'm working mainly on the human heart analysis. So the title of today's presentation is Spatially Resolved Multiomics of Human Cardiac Niches. This is another HCA effort to characterize the human heart using multiomic strategies. And as a background, there was a paper 3 years ago from our predecessors, which utilized single-cell technologies to characterize the human heart, mainly 6 regions of the atria and ventricles. And they found this number of they identified this number of cells and also this number of major cardiac cell types and cell states. But the remaining question was the regions for the conduction system, the cardiac conduction system, which is not in this atlas. So that's why we kind of focused on that critical regions for the heartbeat. And here is the slide for explaining the cardiac conduction system. And it's the signal starts from the sinoatrial node, which is here, and transduce the signal to atrioventricular node, and that signal propagates to the ventricle side with Purkinje fibers and other ventricular conduction systems. And the most important thing is the pacemaker cells, which localize in this two node, SA node and AV node, which haven't been characterized in single cell level in humans. So here is the questions of today's talk. One is, what does the human pacemaker cells look like? And next, by using spatial data, we try to analyze what other cells work together with pacemaker cells in the nodes. And the third one is we made a tool to explore the drugs which directly impact pacemaker cells using single cell data. So I would like to talk these three topics. So first, I will dive into this single cell level analysis of human pacemaker cells. So to do that, we dissect samples from SA node and AV node from the human heart, which cardiac anatomist Professor Yan Ho and my teammate James Cranley contributed a lot for this sample collection. And from these samples, including SA node and AV node, but also other 6 regions from the previous publications, we integrated all the data of the transcriptome and annotate the major clusters such as ventricular cardiomyocytes, atrial cardiomyocytes, and other vasculature cells or immune cells, which becomes the cell number in total more than 600,000 cells and nuclei. And for the conduction system cells, we particularly focused on SA node, AV node, and MPEX. And from the SA node, we could identify a cluster which had the major marker for the pacemaker cells, including these ion channels and transcription factors. And also for the AV node, we identified pacemaker cells and AV bundle cells, which is another conduction system cells. And from the apex, which is one of the ventricular regions, we identified Purkinje cells with these major markers. And the good thing was that we had the spatial data from the same regions. So we utilized cell2 location, which map the single cell data to the spatial BISM data. And we confirmed that the pacemaker cells and other conduction system cells we identified was clearly mapped to the location which cardiac anatomist annotated from the HE image as conduction system regions, such as node and AV bundle and Purkinje cells. So that was a good confirmation that our single cell level annotation is correct. So using this cell type labels, we try to characterize the pacemaker cells, particularly focusing on ion channels and GPCRs, which is known to be important for regulating the pacemaking of the pacemaker cells, including these HCN channels, calcium channels, sodium channels, and potassium channels, which had a unique expression pattern in pacemaker cells compared to other atrial or ventricular cardiomyocytes. Especially this HCN channels is very important to make this fourth phase of the action potential, which gradually automatically increase the resting potential to be able to hit the threshold of firing. And that makes that brings the autonomicity of the pacemaker cells. On the other hand, GPCRs, we identified not only well-known GPCRs, which regulate the pacemaking, but also there were other unknown, previously unknown GPCRs, which potentially can be functional for the pacemaking. We also use attack data. So this study was using 10x genomics multiomics technologies, which can extract not only RNA but also attack data from the same single cell. So we could analyze the gene regulatory network of the pacemaker cells and identified not only activator network but also repressor network from the population. So that was the quick summary of the first part, and I will go into the second part, which is the spatial analysis to analyze what sort of cells localize together with pacemaker cells and potentially work together. So for this analysis, we used BSIM data, both HE image and transcriptome. And for the HE image, we did manual annotation, manual structural annotation with the support from the cardiac anatomist. And for the transcriptome, we used output from the cell2 location and did non-negative matrix factorization to identify groups of co-localizing cells in each factor. And what we further did was that we tried to match the unbiased factors and manually annotated structures to be able to identify invisible cellular niches in each manual structures. For example, in this case, there was a 2 factors matching with this manual structures, which suggest there are 2 niches invisible by the cardiac anatomist. And using this cellular niches, identified cellular niches, we proceeded to cell-phone DB analysis, which we expanded the module tailored to cardiomyocyte interactions by adding the neural GPCR interactions in the module. And this is an example of the analysis. So here we focused on the node structures, which was annotated by the cardiac anatomist. And we found that there was 2 unbiased factors, which matched with this node structures. And that was kind of showing the central part of the node and peripheral part of the node. And also, these are the cell types which localized in each factor, central and peripheral node. And in the central node, there was a pacemaker cells as expected, but also there was activated fibroblast and also glial cells, which was kind of a surprising thing. And in the peripheral node, there were fibroblasts, but also immune cells such as macrophages, neutrophils, DCs, mast cells, suggesting those immune cells promote the fibrosis generation around the central node. So by using this cellular localizations, we did cell-cell interaction analysis using cell-phone DB, and this is one of the examples. We found the transsynaptic interactions, which usually found between presynaptic neurons and postsynaptic neurons, and also associated glial cells. But here we found these interactions mainly between the glial cells, here's the glial cells, and also the pacemaker cells. So this suggests that glial cells and pacemaker cells are interacting with a strong, this sort of adhesion molecules. And so this leads to this hypothesis that in the node, not only neurons and pacemaker cells co-localize, but also glial cells are kind of co-localized together and associating this, supporting this neuron pacemaker cell interactions and potentially regulates the heartbeat. And here is the protein level validation of this co-localization by showing the PLP1 as a glial cell marker and HCN1 as a pacemaker cell marker. So I will go into the third question, which drugs directly impact pacemaker cells. And for that, we developed a tool called Drug to Cell. I think Rasa briefly talked about this last week, so I won't repeat that again. But essentially, this is using the Campbell database, which has the drug and target pairs. And we use this database together with our single cell data to be able to find drugs which target pacemaker cells or find cells in the heart targeted by drugs of interest. So this is the example of the analysis focusing on the pacemaker cells. So here we try to find the drugs which potentially can affect on pacemaker cells stronger than other cells in the heart. So the y-axis is the drug list, which highlighted by this analysis. And it was nice to see there are some drugs which belongs to cardiovascular field, which is already known to affect the heartbeat rate. So this was kind of the positive control for this analysis, but also it was unexpected to see some drugs which belongs to diabetes field. And especially there was a GLP-1 analog, which clinically already known to have a side effect to regulate the heartbeat, but the mechanism was unknown whether it directly affect on the pacemaker cells or affect through the autonomic nervous system. So we validated that using iPSC cardiomyocytes model. So we added GLP-1 to the iPSC cardiomyocytes and analyzed the beating rate with the calcium imaging. And actually this GLP-1 treated, after GLP-1 treatment, the heartbeat, the rate of the firing was increased. So this means GLP-1 increased cardiomyocyte spontaneous firing rate, which supports the finding from the drug to cell. So this is a summary of the talk today. We found unique ion channel GPCR expression profiles from the single cell pacemaker cell data. And we found that pacemaker cell and glial cells are co-locating and interacting in the node. And also, in the end, we found this GLP-1 analogs potentially could be a drug which directly impact on pacemaker cells and regulates the firing rate. So this is the summary of the data set and the analysis, which was published in Nature this year. And the analysis is, main analysis is shown in here. And the module we added to CellPhoneDB is now available at the CellPhoneDB database. And the drug to cell is now available at the TaikiLab GitHub repositories. And all the data set is available at the web portal of the Heart Cell Atlas. So I would like to thank you to all the collaborators, including Sarah and James, who is my teammate, and also Michela Noseda from Imperial College of London. And also I would like to thank the donors and families who made this study happen. Thank you very much.
Thank you very much, Kazumasa. That's a very great presentation and very interesting results. Is there any question from the audience? So maybe I will start by a question. So is it challenging to sequence the heart tissue than other tissue types in the body?
Yes, there are some difference compared to other tissue, I think. One of them is the size of the cells. So the cardiomyocytes in adult, especially in adult, it's sometimes more than 100 micron diameter, and which doesn't go through the chromium flow cell. So it's really hard. I mean, it's impossible to do single cell analysis using adult cardiomyocytes. So people tend to use nuclei rather than cells if you want to use the 10x system. Yeah, that is one challenge. The other challenge we faced is that so when we dissociate the tissue, cardiomyocytes really easy to die. And also there are many debris comes from the after the dissociation, so we had to clean up as much as possible before go to the GEM 10x genomics pipeline. So yeah, that is the, I think, major difficulty of this tissue.
I see. Thank you. Any questions from the audience? You can also type into the chat. Okay, so since we have already reached the time limit, So let me first thank Kazumasa again for joining with us today and sharing his research work. It's great to have you. And also for Lisa to joining us today. This is our final session of this seminar series for single cell genomics. And we hope that all the talks that we had over the last 4 weeks and today's session are helpful for you to gain an understanding of this exciting field of work. And I also would like to thank everyone who were involved and took part in organizing this seminar series, especially Professor Sarah Teichmann and Professor Neelika Malavige from the 2 research labs from Cambridge and Sri Lanka for giving us the support and encouragement to initiate this series and implement it successfully. We had over 150 registrations for this seminar. It was very nice to see that large number. And also we had really great participation so far for the series. So it's great that everyone seems to have taken very good insights on this domain. So thank you so much everyone for joining. And also I would like to thank especially the HCA team, namely Samantha Wine, Lucia Robson, Esther Chua, Yoshi Ando, and Christine for their tremendous support as well for realizing this. And then all the speakers from our Teichmann lab who presented their work. And then, of course, the Malavige lab, everyone from the team members from the Malavige lab as well. So thank you everyone for joining with us today, and wish you all the best in everything you do. Okay, bye.
Bye, thank you.